Hi, hello. This is Syed Ghosh. Welcome to the YouTube Karnataka CET 2021 KCET classes. Yes, this session will start within 5 minutes. Welcome to all the students. Welcome all the students of Unacademy KCET YouTube classes. Still 3 more minutes, then we'll start our class. Let us wait for the people to join. Hello all students. Welcome to the YouTube classes. We'll wait for another 2 minutes. Hi, hello, welcome to all the students to this platform. Welcome to all the students. Yes, students, before going to start the class, so Unacademy has brought you with beautiful plans as per your KCET preparation. So students, in this you can get premium content with affordable price, structured live courses and dedicated live doubt clear sessions, quizzes at regular intervals for self-analysis, batches for complete preparation. So there will be a test series for daily practice test, DPPs, full length marks, etc. with analysis. So for all this thing, only a single subscription. If you get a single subscription, you will get an unlimited access. Students, you can see there is a plans on the screen. So you can avail 1 month, 6 months, 12 months. So there is a comprehensive batch so that you will get topmost lectures from expert educators. So you can avail 10% instead of by using the code GHOUSELIVE, GHOUSELIVE students. So welcome to all the students once again to this beautiful platform for knowledge sharing for the Karnataka CET 2021 aspirants. Welcome to all the students to this platform. You can use the code of GHOUSE. So there will be a comprehensive batch for Karnataka. You can avail it for a very less price. So you can use the code of GHOUSELIVE, GHOUSELIVE. So you can get instant 10% off. So thank you all the students for this YouTube session. So students, today my topic in bio is, as you all know, I have already shared on the screen, biotechnology principles and processes. So in this, students, today my main topic is I'm going to teach Agarose Gel Electrophoresis. I'm going to use Agarose Gel Electrophoresis, students. So that in this, my main topic is Agarose Gel Electrophoresis for this today session, Agarose Gel Electrophoresis. So students, if I start directly with Agarose Gel Electrophoresis, you may have a chance of feeling difficult. So that's why let me give introduction about this Agarose Gel Electrophoresis, where exactly it is going to be. Students, the Agarose Gel Electrophoresis we use in the topic of process of recombinant DNA technology. We use the process of recombinant DNA technology. So in the process of rDNA technology, students, you know, rDNA means recombinant DNA technology. So in this we have so many steps that you can see on your screen. So in this first step, you see recombinant DNA technology involves students, you can see recombinant DNA technology involves different steps. The first step is isolation of DNA. Second step is fragmentation of DNA by restriction endonucleases. Then third one is isolation of desired DNA fragment. Third step is, then fourth one, you can see this isolation of desired DNA fragment. And fifth one is ligation of the DNA fragment to the vector. Then next step is transferring the rDNA host, rDNA into the host. Then the last step in the process of rDNA technology is culturing the host cells in a medium at large scale and extraction of the desired product. Students, let me tell you here, the clarity here is, so students, in this rDNA technology, we are going to multiply the gene product at our commercial purpose. So how much quantity we need and how much product we need to expect, so definitely we are going to get by following these steps. So that step we are calling it as rDNA technology. So in this rDNA technology, the very first step, as I already on the screen, first step is isolation of DNA. Second step is fragmentation of DNA by restriction endonucleases. So after isolating the DNA, we have to restrict by using the enzymes called restriction endonucleases. Means we no need to restrict here, we have to digest this one. So that's the name as given as restriction endonucleases. And the third step, students, the isolation of desired DNA fragment. And the fourth one is ligation of the DNA fragment into a vector and transferring the rDNA into the host. And the fifth one is culturing the host cells in a medium extraction. So students, today we are going to focus most on Agarose Gel Electrophoresis, means separation of DNA. What does mean? It is separation of DNA. Students, here you can see clearly this. In this chapter we are going to learn this one. Students, first of all, after digesting, so we have isolated DNA. So once we isolated DNA, students, then we have to go for restriction digestion. Once you go for restriction digestion, and you have to separate the DNA strands. You have to separate the DNA, because students, there will be so many impurities will be there. There will be so many impurities will be there. So that was the reason why students we use this one. Okay, so you can see this. There will be so many impurities you can observe after the DNA restriction. So once you purify the DNA, is cut into number of fragments by restriction endonucleases. So students, the restriction endonucleases also called as restriction endonucleases is also called as, what is the other name for this? Molecular scissors. Another name for that is molecular scissors. So it cuts the DNA at specific locations. You see the process of cutting the DNA with restriction enzymes. The process of cutting DNA with restriction enzymes is called restriction enzyme digestion. Then once you do this process, and we have to go for incubation, so under optimal conditions. So students, you see this, once you go for restriction digestion, so after that you have to incubate the purified DNA molecules with restriction enzymes at the optimal conditions for the specific use. Okay students, you can see this picture. We have taken DNA sample, this isolated DNA, and then we have added the restriction enzymes to this. So what is the function of restriction enzymes? It cut DNA strands at specific locations. At specific locations, it cuts DNA strands at specific locations, students. Okay, now how can you know how many places the restriction enzyme acted? Yes, and you have to know whether DNA is, because once you isolate the DNA, there will be having so many impurities in that also. They will be having proteins, they will be having some amino acids, so many impurities may also present. So that's the reason why students we use this Agarose Gel Electrophoresis for two purposes. One purpose is what? To know how many fragments are generating. To know how many fragments are generating. To know how many fragments are generating. That is first one. And to purify the DNA. The second step is what? To purify that, because DNA is a negative charge, right? Yes. So electrophoresis means, students, electrophoresis means, as the name itself indicates, phoresis means movement. Phoresis means movement towards electric field. Movements towards electric field is called as electrophoresis. So students, before going to learn into this Agarose Gel Electrophoresis, we need to know what is the use of this Agarose Gel Electrophoresis. Means there is two uses. One, you see, after restriction digestion, you want to know how many places restriction enzyme acted on it. At the same time, when you are giving the electric current here, DNA will get separated, because if some other impurities are present, they may get struck in the agarose layer. Students, you can see this. Agarose Gel Electrophoresis employed to check the progression of the restriction enzyme digestion. Agarose Gel Electrophoresis is employed to check the progression of restriction enzyme digestion. Students, that's what I told you. So you can see, students, the apparatus on your screen. You can see this is an Agarose Gel apparatus. So there it is having agarose gel. You can see this, and this particular lab technician is connecting to the electric field. So once the process get over, guys, see how the process we'll study now. Once the process gets over, then we will go for this type of fragments. So students, here you have to understand number of fragments. You can see this. So based on size. So Agarose Gel Electrophoresis, another step is what? Which is used to know the molecular weight of the fragments also. You can know the molecular weight of the fragments also. You can know the molecular weight of the fragment based on marker. Students, you see here, based on marker. This is a marker, guys. This is a marker. So you see, this is 10 kb, 20 kb, 30, 40, like this is a marker on both the sides. So this is our sample. Students, you see here you see this band you take. What is the band of size of this band means you can compare with the reference one. You can compare with the reference one, so that you can also get to know the size of this one. So you see students, Agarose Gel Electrophoresis is repeated with the vector DNA also. With the vector DNA also. This is also one of the another important point we need to keep in our mind. Then coming to this separation and isolation of DNA fragments. You see students, this is an human double-stranded DNA. So once you taken this human double-stranded DNA and you have used with this restriction nucleases, it cuts the fragments. Okay. So that we are separating. Through which electrophoresis, students? Yes, Agarose Gel Electrophoresis. Through Agarose Gel Electrophoresis. You see students? Yes, these fragments can be separated. Once you digest this, students, you see this. So this double-stranded DNA, once you have used with restriction nucleases, then it liberates and it generates some fragments. So after generating this, okay, what is the technology which you are using here? Which is the method which you are using here? Yes, gel electrophoresis. Students, you can observe here, these fragments can be separated by a technique known as gel electrophoresis. Guys. So let us see now. This is very much important point, students. Since DNA fragments are which charged here? Negatively charged. Students, we have to know this. DNA charge is what? Students, we need to know. DNA is negative charge. Okay, you know that. So why it is a negative charge? Any ideas, guys? Why DNA is negative charge? May I know the answers from you, students? Yes, students, you have to know DNA is an example of what? Nucleotide. DNA is an example of what, guys? Nucleotide. Yes, what is a nucleotide? Just I am recollecting your basics to know that why DNA is negative charge. Because students, without knowing the charge of DNA and without knowing why the DNA is negative charge, then we cannot study the Agarose Gel Electrophoresis. So I want to insist you students to learn the basics of that. Yes, as everyone knows, DNA is a polymer of nucleotide. Yes, DNA is a polymer of nucleotide. Yes, in this also you can see this nucleotide, or you can see there is a nitrogen base plus sugar plus phosphate, sugar plus phosphate, students. Okay, so now you know this is phosphate nothing but what? H3PO4 minus. So because of having the negative charge, so because of negative charge, okay, DNA gets this charge. So because of phosphoric acid, because of which acid, students? Phosphoric acid. Because of phosphoric acid, DNA gets negative charge. So this we have to keep in your mind. You have to keep in mind, students. Okay, hope you all understand this. Yes. So now students, DNA is negative charge means what? Okay, is it anion or cation? You know that is super anion. So students, you see negative end is nothing but cathode and positive end is nothing but anode. Students, in terms of ions, anion is negative charge. Anion is negative charge and cation is positive charge, right? You know that. But in terms of electrodes, students, you can see negative end to positive end, right? Negative end is an anion, right? So that electrode is nothing but cathode. That electrode is nothing but what, students? Cathode. When you give the electricity to that, then what happens, students? Yes, it moves. So on the picture you can see, students, On the pictures you can see here, so this is a size marker. You can see clearly this is a size marker. So in this size marker you can clearly understand what is the difference there. You see, yeah, this is a separation, this is negative charge, this is a positive charge. You can see clearly this. You can get the DNA fragment like this. So you can compare. So this is the DNA bands. This is the DNA bands you get. This is a DNA bands you get after electrophoresis. Which electrophoresis is? Gel electrophoresis. Yes, students, you are understanding now. So this is the principle you need to understand, students. So what is the principle here? It moves from negative end to positive end. So the charge of DNA is negative charge and it is moving from negative end to the positive end. So now students, the most important medium, okay, or matrix. So why we call is an Agarose Gel Electrophoresis? There is an answer is on your screen now. So the most commonly used medium or matrix is agarose, which is natural polymer extracted from seaweeds. The most commonly used medium or matrix is agarose. Agarose, students, this is a, this. So this is a natural polymer. So because of using this as a medium or matrix, so this name we are calling it as Agarose Gel Electrophoresis. The name we are calling it as what, students? Agarose Gel Electrophoresis. Now you have to question and you have to get doubt that what is agarose? Agarose is a natural polymer. Agarose is a agarose is a natural polymer. So from where you can extract this agarose? Yes, from seaweeds. Hope you all understand, students, the basic points here. The basic point here is what? The charge of DNA. The basic point is what, students, here? The charge of DNA. If the charge of DNA we know, then only we can understand. You can say, students, from where you can get this seaweeds. So you see, what are the names of the seaweeds? Also you can see on the screen. The names of the seaweeds are Gracilaria gelidium. Gracilaria and gelidium. Okay, from this seaweeds you can get the agarose. Students, you can see these pictures of Gracilaria and gelidium on the screen. Students, you can observe the picture of Gracilaria and gelidium. These are the pictures, Gracilaria and gelidium. Then students, that once you do this process, the DNA fragments separate or resolve according to their size through the sieving effect provided by the agarose gel. So why we are using agarose gel means it provides effect called sieving effect. It produces an effect called sieving effect. So as I told you already, what is the main application of agarose gel electrophoresis means? It is used to separate the DNA fragments based on the size. Student, here you can see on the screen base pairs. You see 100 base pairs, 200, 300, 400, 500. See, it is the number of base pairs. Suppose if you get a band here, suppose you are seeing this band, so you can easily get to know that the size of the band is how many base pair it is. You know this, 200 base pairs. As you know, length of DNA can be calculated. Length of DNA can be calculated in terms of what? Length of DNA can be calculated in terms of what, students? Base pairs. Yes. So if anybody asks you also, what is the use of agarose? Means, you know, students, it is going to give the sieving effect. You see, students, larger fragment and the smaller fragments. So now also, as I mentioned you also, this separation is happening based on the size, right? So now students, here you created a well. Here you poured your DNA fragments. So now students, you see, now, okay, let us take this side as a, okay, no, you can take this side. So this is an place where you are going to load DNA sample. You are going to load the DNA sample from this side. So students, I told you already, the separation is happening based on the size, right? The separation is happening based on the size, right? Yes. So once the separation is happening based on the size means, as you know, larger DNA fragments will form the band very close to the well. This is called as a well. At the same time, you see smaller DNA fragment, you can see smaller size of the DNA fragment. So they are forming the band far away from the well, far away from the well. Students, you need to understand here is, once you apply this electric field in a medium of an agarose where you loaded your DNA sample, with the DNA sample, then what happens, students? Is DNA is negative charge, so that principle will happen. So what is the principle here? As you know, this principle happens, right? The negative end to positive end, it moves. While it moving, students, while it moving here, you can also see this. Larger fragments, they cannot go far away from the well because of their heavy size, So because of their heavy size, it is going to form the band. It is going to form the band at close to the well. So, and smaller fragments, they form far away from the well. So because of this size, students, you need to keep in mind, so based on the size only, they are going to get separated, and based on the charge also. So that's why Agarose Gel Electrophoresis is based on two important criteria. The first important criteria is the size, and the second important criteria is what? Electric charge. Second important criteria is what? Electric charge. So in this point, what I would like to tell you all the students is, the larger DNA fragment, it cannot move forward, whereas smaller DNA fragment, they can move faster. They can move faster far away from the well, Student, you can see this. You see this, the smaller the fragment size, the farther away it moves from the well. Smaller the fragment size, the farther away it moves from well. So smaller the fragment size, further away moves from the well. So this is the important point you have to keep in your mind, students. The smaller the fragment size, the further away from the well. Student, here, once you separated the DNA fragments, you want to visualize it. Okay, you want to see these fragments, then we have to go for a chemical. So the name of the chemical is called ethidium bromide. It is a carcinogen, student. It is a carcinogen, so we have to take very careful while adding this. So students, it is an intercalating agent too. It is a carcinogenic. Okay, so we have to wear blouses, okay, because it causes cancer also. And then it is an intercalating agent, So the separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide. Fine. So where you have to add this ethidium bromide? Very simple. In the agarose gel only you have to add. Then once this process is over, once 3/4 of the gel, okay, that movement completed, then you have to remove the electricity from that, and then you have to expose to UV radiation. Students, you know what is UV radiation? UV radiation means ultraviolet radiation. What is UV radiation, student? Ultraviolet radiation. So again we have to careful, because both ethidium bromide and UV radiations are harmful to us. So here, see students, we cannot see pure DNA fragments in the visible light without staining. Okay, you may ask, sir, why should we add ethidium bromide? If the question is, sir, why should we add ethidium bromide to the agarose gel, means, as the answer is on the screen that I have mentioned in the bracket item. Student, you can see this. We cannot see pure DNA fragments in the visible light and without staining. Student, you see stained and UV radiated DNA fragments. This is the beauty of the DNA fragments. You can see once the gel. So one point you have to remember is, suppose this is a gel, students, this is a gel, and this is a well, this is a well, and you loaded the DNA sample here. Suppose you form band here, you form band here. You call name A and this band is B. Now my question to you is, which band is having higher size and which band is having lower molecular weight? So as I said you in my previous slide is, so separation is happening only based on the size as well as charge. Okay. So last slide I told you, the smaller the DNA fragments, they move far away from the well. Larger DNA fragment, they cannot move that much far away from the well. So that's the reason why, see, the band A is what? DNA with you can say larger fragment. You can say larger fragment. We can say this is a larger fragment. And this is B, DNA with smaller fragment. DNA with smaller fragment, students. Simple. Then the separated bands of DNA. So once you separate the students that are cut out from the agarose gel and extracted from the gel piece, this step is known as elution. So students, once you subjected to the electric field, so you can get this bands here. So these bands we have to cut and we have to go for a process called elution. Means washing the solvent, washing with the solvent, students. Okay, you see that is nothing but, you see, the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This is known as elution, students. You see the elution, you see what these DNA fragments purified in this way are used in DNA technology by joining them with the cloning vectors. So today, students, my topic is Agarose Gel Electrophoresis. So hope you all understand what is the purpose of this Agarose Gel Electrophoresis, how can we do, and what is the principle involved in that, and how can we give electricity. If we give electricity, based on which principle the DNA fragment moves once the movement done, and then which dye we have to use here, ethidium bromide, then we have to expose to UV light. Once you get exposed to UV light, students, you are going to get these fragments. So the main purpose of this, students, the main purpose is negative end to positive end. Students, DNA is an anion, so it moves towards cation. But in terms of, we are talking in terms of electrodes, means, so negative end is nothing but cathode and positive end is nothing but anode. So our DNA moves from cathode to anode. That's it, student. You can see this once again, all the slides. Separation and isolation of DNA fragments. The fragments can be separated by techniques, nothing but, you can see all the slides once again. So from where this agarose is a natural polymer, and we are going to get from Gracilaria and Gelidium DNA fragments. So smaller will be close to well and larger will be far away from the well. Smaller the fragment size, the farther away moves from the well. Students, we need to use ethidium bromide. So why we have to use ethidium bromide means we cannot see pure DNA fragments in the visible light and without staining. So that's the reason why we are staining with ethidium bromide and we are exposing to UV light. Then you can get the bands like this. So stained and UV radiated DNA fragments. What is this? Stained and UV radiated DNA fragments. So once the process is over, the separated bands of DNA are cut off from the agarose gel and extracted from the gel piece. This step is known as elution. So once you purify the DNA, then we are going to use for ligating with the cloning vector. This is the next step of our DNA technology. Students, so thank you so much for attending YouTube Unacademy session. So thank you guys. Again, we'll meet you in the next sessions. Thank you so much.